GPS mapping, methods of vegetation analyses, cultivation of native flora, and DNA extraction and analyses. And tonight he'll be speaking on cholla with an emphasis on those of Nevada. So everyone, welcome Marc. Thank you.
Are you all seeing our screen?
We got you.
Perfect. All right. Well, then it is.
Am I up?
You're up.
Okay, I just want to warn you guys, this is going to be an informal talk, so if you have any questions, pipe up. Or if you have no heckle, that's fine too. Yeah, my license plate says Chollas. I pull into a gas station and people come up and say, I hate Chollas. So I really think that people are coming in spite of how you might feel about that. Except for one guy said, with a gangbanger, he goes, you know what that is? That's a girl gangbanger. I said, that's a cholla. So how do I,
On the computer there, you can just either click or on the keyboard, just hit the space keyboard.
If you're able to get closer to a microphone, it's hard for us to hear you.
If you can probably just hit this to move, just hit the left button here.
okay.
And then I think they're having trouble hearing you. Is that?
Is there a
can you hear?
microphone?
You can just stand close.
Oh, there, okay.
That's much better.
That's better? Okay.
Yeah.
All right. Oh, I see, there's the,
Maybe not very far.
that'll work. Okay, see if that works. Yeah, so, chollas are nasty. They have these barbed spines that go in very easily, but don't come out very easily. And John Bigelow from the 1856 expedition for the new railroad across the country. If you can't read this, it says, armed with spines worse than those of a porcupine, it is called by the Mexicans chug. The plant is the horror of man and beast. Our mules are as fearful of it as ourselves. So there you go. And the chollas themselves are as dangerous as the roads that you go on to find them too. This is my friend Bob, who rolled his truck when we were out cholla hunting, and he was out ant hunting. And so he went off to get help while I just kind of jacked it up and formed these kind of Dr. Seuss cairns to get it over. And finally we got it over. The Mexicans came and put the chain on it and it fell down. So, okay. So what are chollas? They have succulent cylindrical stems. They have areoles, and I'll show you a picture of those in a minute, with glochids, which everybody loves, and spines. And then the spines are sheathed with retorse barbs. Okay, and here's a spine with the retorse barbs. If they were the other way, they'd be entorse. And there's the areole with the glochid and the spine. They're often woolly in cactus, and there's the sheath and the spine. So those are the characteristics of chollas. There are approximately 35 species with probably more to be described. The last time we went to Baja, we ran into a couple of populations, and I was just like, what? I don't know what these are. So there's always work to be done, it seems like. This is the general distribution. I don't know what these random things are out here. They do occur in Cuba and also in the Dominican Republic, but otherwise they're mostly restricted to the Western North America. This is the one in Cuba, Slender Punchihistrix. I think it's pretty cool. So, and I got this. I have this one growing in Tucson. Okay, what about the evolutionary processes that are important? Apogamy, which is asexual reproduction, hybridization, what everybody knows about. Polyploidy, the duplication of, in this case, whole genomes of chromosomes, and then dioecy. Now, get into these further. Okay, what about apogamy? You're probably all familiar with the teddy bear cholla that drops its joints in there, that's probably how it reproduces. And I really want to start a study that maps these genus, basically these groups of rabbits, which are individuals of the clone, and just see how widespread the clones are, and then map them. This can be a diploid or a triploid. I'm sorry, that's right, yeah, a diploid or a triploid. So we know we have at least two different genus, but there are probably more, we just don't know. In fact, they've done studies like that in European aspen, the Populus trimula, and they have found that they thought that the clones were much bigger than they were, but it turned out the clones are only maybe 3 or 4 rabbits each. So they've also done some work on Populus tremuloides, our aspen, but they haven't really mapped them. I want to make a map of clones, so we're starting on that project. And the success of these apomictic species may be because they isolate these genomes that might have a particularly good adaptation to the environment. It's almost like science fiction where you develop a superior soldier and then you clone them to have a superior army. So I don't know if that makes any sense, but this is the one that's in Baja that looks like Bigelovii, it's Cerebae. And you see it dropping its joints in the ground and they're starting to grow, and that's primarily how they reproduce. Okay, then we have hybridization, which can occur from the smallest stem species. This is Christmas cholla over here, which you can't see very well. It has a very thin stem that hybridizes with the fattest chollas. Of course, hybridization is a combination of genetic material between or among species. And in chollas, the F1 hybrids are generally morphologically evenly intermediate. So you can usually guess when you see a hybrid. And these are the hybrids that we've hypothesized so far. Until they're actually proven, they're called putative hybrids. But the thing is, though, even though they're not considered proven until after, well-designed genetic studies or cytogenetic, most cholla hybrids are evidenced again by the morphology and then the occurrence of maybe just one individual among hundreds, if not thousands, of the parents' individuals. So if you have one that's intermediate, it's obviously a hybrid. Don't need this thing to prove it. Okay. Here's one that we have in Nevada, a hybrid. It's between Echinocarpa. I'm terrible with common names. Anybody know?
Silver cholla?
What's that?
Silver cholla?
Silver, yeah, either silver or
Golden.
depending on what color it is. It's generally silver in Nevada. And then Acanthocarpa, which is
Buckhorn.
what's it? Buckhorn. Okay, that's right. Anyway, now you'll see hundreds of either the species. Every once in a while, you'll come across one and you'll go, is that Acanthocarpa or is that Echinocarpa? And that's, it's probably have the hybrid, in which has been named hybrid deserta. Then we have polyploidy, which is the duplication of chromosomes. And it's very important in chollas and other cacti as well. I think this one's a hexaploid, 33 chromosomes on one side and 33 on the other. If it was a diploid, it'd be 11 and 11. And so far, especially in chollas, we have not seen any aneuploids. We've counted hundreds of thousands of these things and never seen any. So we assume that aneuploidy is lethal. Aneuploidy is when you have just a couple of extra chromosomes, whereas euploidy, you have the whole set. So in the Chollas, you have euploidy. So it could be 22, 33, 44, 55, 66. I think there's 77, but I can't remember. Okay. And it's a similar hypothesis for the success of these polyploids is you basically, it isolates these particularly adaptive sets of genes, and it keeps them from back crossing. So you basically they stay the same, almost like a clone would do the same. All right. Here's an example of this is Ramosissima, which we were just talking about. It's called the pencil cholla or the diamond cholla, which occurs in Nevada, and down in Arizona. It has some tetraploids, and the rest of it so far has been diploid, including the ones in Baja California. And I grow those in the common garden, and I look at them, and I look at the flowers, and I look at the number of spines, but I haven't seen any morphological correlation between the diploids and the tetraploid. So without morphology, I don't want to name it. It's kind of, because you never know, you can get a spontaneous polyploid in any population, and it doesn't make a species all of a sudden. So it has to evolve, but the polyploidy does isolate the genes, makes the evolution quicker. All right, and here's slender punctured whippley-eye, or the whipple cholla. It's an easy one. It's diploid in the eastern part of its range, and there are two tetraploid forms. One's just north of our house in Arizona, near the Walpi Reservation, anyway. And this Walpiensis looks a lot like the diploid. So the hypothesis is that this one probably came from this one and then evolved slowly into this other one, this genus, which is adapted to more drier environments, so it's become more diminutive. Yeah, this is the distribution. So you have the diploid whippley-eye coming up into the data, and that's it. And then the notice versus the tetraploid forms, this doesn't quite make it into the data. And this is multigeniculated, by the way. That's its distribution. Sort of. Yeah, that's it. Okay. There's whippley-eye, the one that's in Nevada. You can see it's fairly spiny, big fat stems, more of a bush. And there's Enotus, which almost smooth fruits. And then I was out after I'd already published on these damn things and separated them on various characters. And from the common garden, I noticed that the Enotus has these really thick fruit walls compared to those of the whippley-eye. It would have been a nice character to use, but unfortunately, I didn't find it in time. But it also shows you the importance of doing common garden experiments. So triploids may have punctuated evolution, and this is just my own hypothesis. So if you think it's nuts, then that's okay. But here it is at meiosis. This is a triploid. There's a trivalent right there. It has 3 separate little chromosomes. And so that means when they separate, 2 can go that way and 1 can go that way. Okay. Now, here's where the punctuated stuff comes in. If you have a cell that has 2 chromosomes consistently going to one pole and 1 going to the other, you can basically have a viable euploid cell then. But that only happens every once or two to 3,000 times. So it's very rare. So they can hypothetically reproduce, but rarely, and that's where the punctuated hypothesis comes in. And again, like I've said more than once, that these races that are mostly apomictic can preserve this particularly adaptive genome. Next, we have dioece, in which you have basically dioece means two houses. I learned that in biology. So one plant will have all pollen sterile flowers, basically those are the girls, and then one plant, another plant, separate plant will have the pollen fertile flowers, which is basically the boys. Okay, this is an example. In fact, we weren't talking about this, we're talking about another species, but this is Yellow-Piensis, and I included this because it has such nice examples of dioecious flowers. And here you can see the anthers are just not developed at all, and so there's no pollen. But the stigma, man, they're beautiful. And here you have anthers with copious pollen, and then the anthers are kind of funky. So, yeah. We recently described a new cholla. What was that from? California? Anyway, I think we were doing surveys for some clearance, and every morning the UXO people, the unexploded ordnance experts, had to say, don't touch it if you don't know what it is, or if it looks like a bomb. And so one day they had off. And so we couldn't work because we had to be there and we had to have that lecture every single morning. So we drove out, we were going to look at the reference population for slender bunch of Munzii. We passed one of these guys, and I jumped out of the car and the truck, and I looked at it, and I looked up, and I said, this is a new species. And of course, all these people in these trucks are kind of rolling their eyes. Yeah, right. Turns out it was, because it looks a lot like Acanthocarpa. I mean, Echinocharpa. They were saying, that's just Echinocharpa. Well, it's a hexaploid. And it turns out that there's this correlation between hexaploidy, variable flower color, and dioece. And so we call them the sexy hexies. And it made the front cover of Cactus and Succulent Journal, cover girl. But yeah, so it has extremely variable flower color, all the way from yellow to almost a red. And then the anthers are variable too. Pretty cool. And then we started looking around, and there are at least three others. Calmaliana, which is Baja California, Syntholipensis, Baja California, and Wolfii, which is Southern California. And they all have these three characteristics: hexaploidy, dioece, and flowers of variable color. And so, yeah. So if you have a hypothesis as to why this is, that's like to hear it. One working hypothesis is that, okay, these hexaploids have to come from diploid originally. So when they do develop, they're very isolated. And so when they start to reproduce, they're almost like clones because they're very limited. So in order to kind of combat this low genetic diversity, they evolve into outcrossing, which enables them to spread their genes a little bit more. Okay, what about the Nevada slender puncher? We talked about this one a number of times. This is the teddy bear cholla, the slender puncher bigelowii, named after J.M. Bigelow on that famous expedition. It's, this is Acanthocarpa. What you have here in Nevada is subspecies of Acanthocarpa. And I did a study on Acanthocarpa, its variation within the species. And I did multivariate analysis of the morphological characters. And what you get out of it in MANOVA is a really cool table. It's called selected homogeneous subsets, which is a mouthful. But these numbers refer to the different taxa. And you can see that like stem diameter might be a really good thing to put in your key. Tubercle length is obviously a super good character because these are all statistically different if they're in a different column. So, so this makes it super easy to make up a key if you have the data. So here you go. I might add too, when you're trying to identify a group of within a group of difficult species, it's always good to look at 2 or 3 plants, or maybe more, to get kind of an average. Because that's what we're talking about. All the numbers that go into this chart are all going to be like the average height is 83 centimeters here. So. And also in Arizona, we have this subspecies Ramosa. Another thing too that's nice about multivariate analysis is the discriminant analysis, or discriminant function analysis, which gives you a percentage of correct classification. So just through doing this over and over again, we know that among species, the correct classification is generally about 99%. So that means 99 out of every 100 plants you measure are going to be right in the right species. And generally with subspecies, we want to look at about 95%. Unfortunately, this one only had like 85. So I said, I don't know if I want to recognize it, but my co-author said, it's a good one. I said, okay, whatever. So, and this is the third one, which is a lot different looking with fewer spines. Okay. All right, here's the Bigelovii again. Echinocarbon with a very spiny fruit, generally whitish spines. This makes it the silver cholla until it's not, then sometimes it has golden spines. But generally it's the spine sheaths that are golden, not so much the spines themselves. And then the multi-geniculata, which has been looked at over and over and over again. Which is it's very, very compact. And Dr. Pinkava, my advisor at Arizona State University, said, in fact, I think he even published it in the Flora of North America as probably a hybrid between the Echinocarpa and Whipplei. And my data, even my morphology data, did not show that. I was just kind of irritated over that. But, just recently, I think we were just discussing it with somebody. We found it up in the Las Vegas range, and we found a whole bunch down here that are jungle heat. I don't know how many. I did a status report for it way back in the day, and back when you were all in kindergarten or something. But anyway. I forget how many thousands I said there were, but this population increased like the number of individuals by like tenfold. Another interesting thing about this species is down near the border, Tom Van Devender did some pack rat nidding studies and identified one of this thing down here as Whipplei. I thought, wow, that's cool. So to U of A and I looked up this material and I said, oh, that's not Whipplei, it has too many spines. It's multigeniculata. So back 14,000 years ago, we had multigeniculata down here with stuff like Joshua Tree and all that kind of stuff. So that was kind of cool, I thought. And so a friend of mine found a population on Harkuvar Mountain, which is, anyway, it's down here someplace, maybe just there, but doesn't look right. I don't know where the key is. It's probably behind here. Anyway, it was about halfway down between these populations and these populations. The weird thing is, though, is that this population had almost spineless fruit, which matched the Nevada populations. The Arizona populations of multigeniculata have spiny fruit. So this came from up here and not from the closest one, which is kind of cool. So it is obviously a remnant. And this Ramosissima, which we were just talking about, is the diamond cholla, which it can be confused with the blue diamond cholla, I guess, and the pencil cholla. And we talked about Whipplei, and I just wanted to throw in a notice in here to show you it's more diminutive form. Okay. And so we have the Pinkava hypothesis that multigeniculata is a hybrid between Echinocarpa and Whipplei. And if you go to a multigeniculated population, there's often Acanthocarpa. And then every once in a while, you'll see a hybrid between a little multigeniculate and Acanthocarpa, and it looks just like an Echinocarpa. And my hypothesis was that Echinocarpa originally came from a hybrid between multigeniculata and Acanthocarpa. I was excited about that. So, first of all, Pinkava was dead wrong. As we can see, where's our, I think I'm too close to it. There we go. Yeah, multigeniculata just comes out right next to Whipplei. It has nothing to do with Echinocarpa. So, and then also Echinocarpa obviously is not a hybrid between Acanthocarpa and multigeniculata. So we were both wrong on that. So. All right, what about these so-called chollas that ain't Cylindropuntia? One in Nevada is the club cholla, Grossonia peritiorm, and forms these shin dagger kind of clones out in the desert. And there they are. They've got a little bit of spiny fruits. There's the flower for that one. And the other one that they often call cholla is the Micropuntia pulchella, which has a big tuberous root. And here it is in fruit. And it has a nice, I think, a really pretty flower. They're not that hard to grow. It seems like they're easier to grow in pots than they are on the ground. Maybe it's because the gophers can get to the roots. That's all I have to say about chollas. Do you have any questions?
Where's this rose, that one? Yeah.
That particular one, I don't remember. Almost all my pictures are data-based. I could look it up.
So where are they mostly found?
Oh, where are they mostly found? Shoot. Oh. Yeah, the low-lying areas in northwestern Arizona, southern Nevada. You get into California, I think.
I photographed it north of up in the not Basin, yeah, Basin Range National Monument. You know where
Okay,
it is, Basin Range?
Basin.
What, the place where we just worked? Yeah, Barnum, Basin Range National Monument.
Where's that?
North of Brannigan, that
Okay,
way.
okay.
Yeah, Mount Irish, I couldn't think of yeah, I photographed it up there a couple years
Okay,
ago. on a
very
file.
nice. I mean, if it's not in flower, it's really, it's very hard to find. Any other questions?
We have questions up here in Reno too, when you're ready.
Yeah, so I was gonna say, speaking of Basin Range, there's a possible multigenicular population up there too that's on iNaturalist,
Oh,
which is kind of like pretty far north for it
Huh.
to be. But if you want to look that up, it was Jim Boone saw that and told me about it. But yeah, it just sort of seemed outside of its characteristic range. It was pretty far
Okay.
north.
has it been vouchered yet?
I don't know that it has. You know, it looks to me like it's multigeniculata,
Okay,
but it's pretty far up there.
I don't know what else it'd be. The Carpa goes pretty far north, and it gets very diminutive.
Or this one look, I mean, it just looked like multigeniculata,
Yeah.
but I haven't seen it in person.
But that seems even too far north for any kind of Carpa.
They said they had some questions on that.
Yeah, we want
Oh.
to take the questions from Northern Nevada. I guess
Yeah,
we can-
hi there. My name is John Weiser, and I raise a lot of the cactus that you were showing. The Grussonia puchella, the most northern population I've ever seen was along the eastern shore of the Black Rock Desert, in the foothills to the east of the desert itself. And there's quite a population up there. The other place I've seen it really big population and dense was the Tiobe Range, on the east side of the Tiobe Range, where the alluvial fans peter out into the Big Smoky Valley. There are literally thousands and thousands of them up there. We also see it at Churchill Narrows, around the Carson, Crystal Springs area. So it is around, it's very sparse populations in the western part of Washoe County, but it is around. And I do raise it, and it does have a super large tuber, probably two grapefruit size tuber under the ground. It does take years to establish a little cutting or a seedling. It does take many years to get it to establish and turn into a big plant. But yeah, that's the northernmost I've seen it. Was in along the Black Rock Desert on the east side in the foothills. So I just
Is this all in Nevada?
yes,
Okay,
Northwest.
yep.
very cool. Yeah, you can take the little stem segments and plant them, even though they have the tubers. Takes a long time. I've had some in the pot now for 2 or 3 years, and they haven't changed much.
Yeah.
All right, any more questions?
Questions in the chat from George as well. And he asked, what's the northernmost range of cholla in Nevada, and how many species does Nevada have?
Well, I told you all the species. You have to count them yourself. And the northernmost, I could not tell you. I'd have to go look in the one of the herbaria to find that out, or sign that or something.
Mark, this is Jerry.
Hi, Jerry. Have
How
we
you
actually
doing?
met before?
Haven't seen you for a while.
Yeah.
So Micropuntia pulchella is the furthest north one I know, and it gets into Humboldt County,
Okay.
south end of that. But just as a distribution, here's what we have for counties in my new book, right? We can use it. Churchill, Elko, Esmeralda, Humboldt, Lander, Lincoln, Lyon, Nye, Persian, Story, Washoe, and White Pine. So it's also in southern Elko County, which would be as far north as Humboldt County.
Okay. As long as you're looking at the book, are there any choices in this for Nevada?
Not that I know of. Let me just look. Let's see. Nope, you got them all.
Wow.
How about that? Okay, I'm.
Thank you, Jerry. No, that's good. The book is useful.
Yeah.
All right. And then George's last question was, would you agree that the diversity hotspot is in Baja?
Yes, I would agree on that 100%. Yeah.
Yeah, that makes sense. I have a question. What do you wear when you're doing all of this field work?
Sometimes nothing.
Oh.
That seems
Depending on
dangerous.
who's around. Yeah, generally shorts and a t-shirt.
Yeah,
But sandals are not a good idea.
yeah. Fair enough. Thank you. I think that's it from us up here.
Anyone else down here?
What is your favorite cholla?
It might be the teddy bear. Is this the meanest?
It is mean.
Yeah, I had a cactus garden when I was 14, living on China Lake. And they still had civilians on the base, China Lake Naval Weapons Center, what it's called. I remember carrying one of those and dropping it on my bare foot. Yeah, taking that thing out, that was a lasting impression on me. And they're just so cool, the clones and the ploidy and all that. It's very cool.
Can you speak a little bit more to how you talked about how even in clonal there's variations among clones? I'm not a botanist. I'm just a gardener. Can you explain a little bit more how that happens with cholla? Like how clonal populations start to diverge among themselves? Is that what you're saying?
Yeah. Remember that slide I had up there with the chromosomes going every which
Yeah.
way? There's a possibility that the diploids came from triploids. Because if you have 22 chromosomes going to one side and 11 going to the other, then if 2 of those gametes with 11 chromosomes get together, now you have a diploid again. So what we're finding is you would think that the diploids would be more fertile. But we're not really finding that. So, but it would explain it if it was a secondary diploid, maybe that's why it's still not fertile. So we know there are at least 2 different genetic types in the teddy bears. But beyond that, we don't know. For all we know, all of the triploids are one genetic clone. We just don't know. So what I want to do, and I've discussed this with more than one geneticist, and what I'd like to do is start out and do kind of a broad sweep of the teddy bear chollas and get maybe 30 samples throughout their range of both the diploids and the triploids, and see if we find differences in their genetic makeup. If we do, then we can start doing maybe focusing on those areas that have more genetic and see if we can pin down and actually map how big the clones are. So on that populus tremula, the European aspen, they were finding that the clones are just made up of just a few individuals. And they expected like the whole hillside to be one big giant clone, but it wasn't the case. So we don't know yet. We need to do more work. There's always more work.
It seems like because of the way they spread, you could have clones that are clear. I mean, like you could imagine it getting stuck on an animal or something.
Yeah.
And you deposit somewhere else. It seems like your job's gonna be harder than the aspens because they're all just there. And they just move the dirt.
They clone from rhizomes. So, and these obviously they could be carried for miles and miles. So So we don't know. We just have to, we have to do the work to figure that out. So there's,
Is there any value of having these variation in color of the flowers that, for example, would they attract different pollinators, and could that mess up your whole idea about sort of selfing, given that they can move pollen around? And I mean, how does that factor into the story?
that's, as an ecologist, that'd be something you could explore. I mean, we know that different colors of flowers attract different pollinators. But how these variable flower colors evolve, especially within these hexaploid species, like how did that happen? You know, what was the selection pressure for this different? Just I'm clueless.
Yeah.
You know, except that maybe you do get more of a variety of pollinators to cross-pollinate. But if that were true, then all plants would do that. All plants don't do that. So, yeah. So basically, I'm back to ground zero. I don't know.
I think there's another question that came up.
Yeah. Is there a question from far?
Look it up.
Popped up.
See. Says, what is the ecological role of cholla play in the Mojave Desert near Las Vegas?
Ecological. Again, here's the ecologist. See, what kind of ecological role? It seems like that they're there because that's what they're adapted to. As far as like animal ecology or insect ecology, pack rats, there's an association. So they like to make their nests out of cholla stems. That's the most apparent. They're also being so abundant, they exclude other species sometimes, especially within those clones. The big teddy bear cholla, that's all you see. You don't see many other shrubs in there. So
They provide cover for a lot of things.
Yeah.
Because who's going to chase them?
yeah. So,
Bird nests.
yeah. You think of any other ecological?
Bird nests, yeah.
Lots of birds nest in them because then
Oh,
they
bird
don't get
nests.
predators. Yeah.
Very good. Yeah, that's a good bird. Very common to have bird nests in there.
Right, till that looks gross, so they'll hang out underneath them.
And eat the seeds.
Yeah.
Fruit.
And there's the cactus wren.
I was just saying cactus wrens, they say ouch, 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 ouch for a reason.
Yeah. I actually did a paper on the cactus wren in zoology back like 1970 or something. But they don't make the nest, if I remember. Some other bird makes the nest, and they just take it over.
Another question.
Another question?
Could you please talk about the stem attachment? Some chollas jump, but some don't.
Basically, it's just a lack of attachment. You don't have much vascular material and crossing from one stem to another, and like in the teddy bear cholla. So they just break off very easily. There's not really an abscission zone. I think, again, I think it's just a lack of thick vascular bundles that are, they're just not crossing that point of attachment.
I have a question. In the apomictic species, they're still producing nectar, presumably, right? And
Uh-huh.
pollen and everything like that.
Uh-huh.
Okay, so they're still getting pollinated. They're just not, setting seed or fruit, right?
You're talking about the dioecious?
In the apomictic species.
Apomictic.
Yeah.
Well, I think the apomictic species can also have seeds. You can still get seeds from the fruits of teddy bear cholla. And they're viable. But obviously their main mode of reproduction is just dropping off joints.
Sure.
So, and again, it kind of goes back to the punctuated evolution, especially in the triploids, where you might get just a few fertile seeds out of a whole population in 1 year. And what are the chances of those seeds actually making it someplace else and forming a new clone?
Sure.
Yeah, genetic clones. So, yeah.
I think that's all the questions we have. I don't see any more in the chat. What about up north? Okay. Well, I think we can thank Dr. Baker one more time. Thank you. And thank you, everyone. I think that is all for tonight.
Yes, thank you, everybody.